Hello, everybody. Today I'm going to talk to you about next-generation sequencing-based analysis of hematological malignancies. My name is Christian Thiede, and I'm working in the medical department, the first medical department of the University Hospital in Dresden. I've been involved in hematological malignancies for the last 20 years, and since 4 years we are working using next-generation sequencing. And today I'm going to share some of our experience on these platforms. The next slide is my disclaimer slide, just showing that all the data and opinions I'm presenting to you today are solely my own and are not Illumina's opinion. As you might know, assessment of hematologic malignancies is a very complex process involving several subdisciplines. Of course, still morphology is the primary entry point to diagnose a hematologic malignancy, but more comprehensive assessment of these diseases involves flow cytometry as well as cytogenetics, and more and more also molecular technologies have been used in the past years to detect individual alterations. But with the growing number of molecular changes diagnosed during the last years, other technologies, especially next-generation sequencing, have become much more important. And today I'm going to talk to you about this. In AML and also other diseases, the use of next-generation sequencing has revealed a variety of molecular alterations being present in patients. So this is the molecular landscape of patients with AML, generated on the basis of more than 200 patients with acute myeloid leukemia based on whole genome or exome sequencing. And as you can see, there is a variety of molecular alterations. This only depicts the more frequent ones. And a typical AML patient carries about 12 to 15 mutations in genes, and 4 to 5 of these changes are recurrent. And a very similar picture can be drawn when you look at Myelodysplastic syndrome. Here, as you can see, the tail is much longer, so the individual changes are present in only very few patients, but the list is very comprehensive. And the number of changes being present per patient is only about 3. So you see that the detection of these alterations becomes much more important, not only for the research part of these diseases, but also for classifying the disease. And the most recent release of the WHO classification released last year already carries several only molecularly defined alterations in the list of changes being used for the classification of patients with AML, namely patients with mutant NPM1 as well as biallelic mutations of CEBPA-alpha, and also a provisional entity defined by the presence of mutations in the RUNX1 gene. And this is not only the stopping point. More recently, the new ELAN classification of the European Leukemia Network, published by Dönert et al. 2017, also involves a comprehensive list of mutations in genes like NPM1, FLT3, or CEBPA-alpha, and a number of novel alterations like RUNX1, A6L1, or TP53 to define individual risk of patients to respond to treatment with chemotherapy. And this clearly shows that we do need more comprehensive platforms to identify these alterations. And as you can see on this slide, in the near future, the classification might become even much more comprehensive, as shown by the publication by the Sanger Institute in cooperation with the AML CG in Germany. Using next-generation sequencing-based typing of patients with AML, you can already identify a number of individual subgroups which are clearly represented by individual mutations, as shown on this side of the table, like nucleophosphamide-1, NPM1, CEBPA-alpha, but also much more rare alterations like the IDH2 arginine 172 mutation. And this also translates into very different prognostic information which can be obtained. So patients with biallelic CEBPA-alpha mutations have a much more favorable outcome compared to patients with TP53 mutations being aneuploid. And this is also important not only for prognostication, but we do have a growing list of alterations which do respond to specific treatments. FLT3 ITD mutations or TKD mutations are known to be responsive to treatment with tyrosine kinase inhibitors like sorafenib, midostaurin, or quizartinib. But we do have also novel alterations like the IDH1 or 2 mutations which are responsive to inhibition using specific drugs, and there is a growing number of these drugs being developed presently. So the comprehensive classification, molecular classification of hematologic malignancies becomes of growing importance for the clinical management of patients. Here you can see what happens when you use these drugs in patients. You can substantially improve outcome as exemplified in the RATIFY study, a large international study where more than 3,000 patients with AML were typed for FLT3 ITD and TKD mutations and then were randomized to receive either midostaurin or placebo, and you can see the addition of midostaurin to treatment significantly prolonged survival and increased the probability of overall survival in this patient population. So we do need next-generation-based technologies, especially to improve classification and prognostication, but also to identify targets for treatment. And there is a growing body of evidence that it might be also important to use this technology for the detection of minimal or measurable residual disease in the near future. And there is an increasing number of reports on this in the literature. Today I will share with you our experience using the Illumina platform, and most of the data we have generated so far are based on the so-called TrueSight Myeloid Panel, a comprehensive panel involving the typing of 54 genes, 15 of these complete, and in 39 genes there are hotspots regions which are covered. In total, this assay involves 141 kilobase pairs of sequence, which can be amplified in parallel and then analyzed. We have used this on more than 1,200 patients with AML in different questions and different research projects, and also starting to use this in the diagnostic field. What I'm not going to share with you our research results because most of the data are not published so far, but what I want to share with you is our experience in trying to standardize the method and also some of the issues we saw, some of the problems we saw in the application of the panel. To start with the approach to standardize this method, what you can see here is an approach where we wanted to evaluate the ability of next-generation sequencing to type AML patients or AML samples. And we found a consortium of 11 laboratories around Europe which were involved. About half of these groups had experience with the platform. The other half of the groups were not that experienced when they started to use this or started on this consortium. And we generated a pool of samples, which were 12 clinical samples, patient-derived samples with known variants, a commercial control DNA, and a cell line DNA and normal DNA samples. We then split these in 2 runs containing 8 samples, and used the 150 base pair paired end sequencing chemistry. Single nucleotide variants, or SNVs or InDels, were then called using the Variant Studio software, and we compared the results for the SNV calling accuracy, the variant allele frequency, and also the reproducibility of the results. We first started to look at the commercial sample, which is a cell line which can be bought with containing alterations with a dedicated variant allele frequency. And as you can see on this slide, we were really delighted to see that there was a high concordance in the found variant allele frequency to the actual variant allele frequency depicted over the individual columns. So there was a very high concordance of the results with the individual groups. We then went on to look on the clinical samples and were somewhat surprised to see that there was a major difference to the results obtained with the commercial sample. As you can see here, only for those sampled with a high variant allele frequency, there was a considerable concordance between the individual groups to report these alterations. So only when the variant allele frequency was 40%, about 50% of the groups reported this change. Whereas the changes with a lower variant allele frequency were reported only in single laboratories or in a considerable lower number of labs. The reason for this was a combination, when we looked at it in more detail, a combination of coverage and low variant allele frequency. So especially changes with low coverage and a very low variant allele frequency were likely to be reported, underreported in this comparison. When we went on to look on in a more detailed fashion, we I want to share with you our experience on the different platforms, and we have so far used the Myeloid Panel on all 4 major available platforms so far from Illumina. The majority of runs were done on the MySeq instrument. We do have some experience also on the MiniSeq instrument, which is probably one of the newest in this family. We do have a major experience on the NextSeq platform, and we did some runs also on the HiSeq platform. When you compare all these 4 platforms, we have to see that especially the MySeq is very well suited. In our experience, also the MiniSeq works very well, and the NextSeq, but the HiSeq results were somewhat suboptimal, and I will come to that back during my talk. When we look for the coverage, the results were very reproducible, and you can see here that for 97% of the amplicons in the panel, we get a more than 500-fold coverage. And this is true on all 3 major platforms where we do the majority of the runs on. So the results are assay-specific and not platform-specific. But we do see also major differences. So when you look at this, you see that the majority is covered quite nicely. But if you look in more details, see that there are dropouts where on all platforms these assays underperform, and you do see really suboptimal coverage in certain areas. And this, in fact, is represented by GC-rich areas. For example, one example is CEBPA-alpha, which is an extremely GC-rich gene where we do have major problems in covering this alteration. There's also a impact of the low coverage and low variant allele frequency in detection of false positive results. When you see at this or look at this, we do have a low confirmation rate shown in red in those changes where the mutational load is below 10%. And this is especially true when you combine this with a low coverage, so especially changes with a coverage below 50 reads per alteration there is a very high non-confirmation rate. So when you look at your results, it's a very good idea to have a sufficient coverage and to repeat results if you do see suboptimal coverage of your results. We do have also some evidence that there is an issue with the DNA quality. We were not aware of that before. Of course, it is known for a while that paraffin-embedded tissues, for example, generate a low-quality DNA, but this is DNA which was generated from fresh material from cells using a standard assay. It was stored for a while, and you can see that samples which were stored for about 20 years, that date back to 1997, have a much higher background rate compared to samples which were more recent. So there is some evidence, we have not formally studied that in detail, that the aging of DNA also increases the background, and this might also be an aspect where you want to look at when you do see an increased number of background results. Another aspect where we do have seen problems is the detection of InDel mutations. One example already mentioned is the CEBPA-alpha gene, an extremely GC-rich gene with GC content up to 83% in certain regions. And the mutations here are typically InDel mutations, which are on average 1 to 10 base pairs long, but can be up to 75 base pairs. And you see here one example in the fragment analysis. This is the wild type signal, and this is the mutant signal, which is longer than the wild type signal. And you see this very easily due to the frame shift in Sanger sequencing. But when we looked at our results using different mapping and analysis algorithms, using the 2 times 150 base pair chemistry, but also the 2 times 200 base pair analysis, we did have problems in detection of these InDel mutations. We only consistently detect these when we use the PINDEL algorithm on these samples. So when you're looking at InDel mutations, especially the longer ones, and here's a number of genes which are frequently present in myeloid malignancies, mutated myeloid malignancies, flat-free mutations, colorectal mutations, RUNX1, WT1, CEBPA-alpha, also in KIT or in DNMT3A, you can see these alterations, and it might be difficult to detect them. You have to be aware that you can have underrepresentation of these mutations due to the fact that they are not detected. What do we plan to improve the performance of the Myeloid Panel? So there is an updated content during the last 4 years since the first version of the Myeloid Panel was, or TrueSight Myeloid Kit was released. A number of novel genes have been published which are relevant for myeloid diseases. We want to eliminate some of the not so important content currently involved, and we want to have different content for research and diagnostics, to have a smaller kit for diagnostics and a more comprehensive one for research purposes. We want to optimize, and when I say we, I say Illumina, wants to optimize the chemistry, and we also want to use improved algorithms for the detection. An aspect which is important for Germany. In Germany, next-generation sequencing was banned for quite a while, but nowadays we can use next-generation sequencing, and there is a growing list of diseases where we do want to use this technology, especially in, of course, in myeloid malignancies like AML, MDS, or myeloproliferative diseases, but also in CLL, TP53 mutations have been associated with dismal outcome, and patients might be eligible for novel treatment strategies. And also for the patients with myeloproliferative diseases, A601 mutations might be an important prognostic marker. Another evidence or another aspect one has to have in mind when using next-generation sequencing in patients with myeloid diseases is the fact that there is growing evidence that the presence of a mutation not always gives you evidence for disease. So in the absence of other clearly defined evidence of disease, the sheer detection of a mutation does not hold the potential to really diagnose a disease. And this is exemplified here based on the data published in the New England Journal in 2014, where up to 10% of individuals between 70 and 80 years of age have detectable somatic mutations in the circulating blood cells using next-generation sequencing. And this is not disease. This is so-called CHIP or clonal hematopoiesis with indeterminate potential. So in the absence of definitive morphological evidence of a hematological disease, these alterations are not indicating the presence of a hematologic malignancy and should be just kept in mind. This can be associated with very normal outcome and is only representative for disease if there is additional evidence based on morphology or other biomarkers that there is a disease present. So in summary, I think next-generation sequencing in myeloid diseases, it is a very important platform technology to identify novel alterations. The use of multiplexed amplicon-based strategies is a very powerful strategy because we can detect many alterations in parallel. And this is especially true in patients with AML, MDS, and myeloproliferative diseases, where it holds great potential for very comprehensive molecular diagnostics. But we do need optimization for the content which we want to detect, the performance in particular genes, especially for those which have high GC content, and those genes where we do expect large InDel mutations. And we'd have to deal with the unspecific low-level variants. This has to be kept in mind, not only technically, but also biologically for the patients with the CHIP detectable in the blood. And with that, I would like to thank the people in my lab doing the majority of the work I showed to you today, the Study Alliance Leukemia, which provided the majority of the samples, and the fruitful collaboration with several institutions within our faculty, but also outside our faculty. Thank you for your attention.